Todo listo.
Good day to everybody. Thank you for joining us to this fifth section of the Symposium on the Proboscideans in the world that the Seminario Relaciones, Hombre-Fauna is co-hosting with other institutions. And here is also my colleague and co-director of the Seminario, Eduardo Corona. Today we have really an important guest who has been supporting much in the studies in Mexico, mostly in regard to the topic he's going to talk about. Dr. Florent Rivals. He's researcher in the field of paleoecology on Pleistocene deposits. He got his PhD on prehistory in the year 2002 in the University of Perpignan in France. In 2004, he did a postdoc at the American Museum of Natural History, and in 2005, he got a Humboldt Foundation scholarship to do research at the University of Hamburg in Germany. Between 2007 and 2012, he have a junior research position at the Institut Català de Paleoecologia, Humana y Evolución Social in Tarragona, and in 2013, since then he has he's a professor at research professor at ICREA from the same institution. Presently, he's main researcher on the project research on paleoecology in archaeological context of the Upper Pleistocene that is funded by the Spanish Minister of Science and Innovation. His research focuses on evolutive paleoecology with an emphasis on the influence on the climatic change and the adaptation of the mammals, particularly Proboscideans, echids, and cervids. He uses much techniques like microwear and mesowear wasting in the molars, and also he follows a multi-proxy approach to get the best knowledge on the diet and the landscape where the animals used to live. He also does studies in past habitats and try to learn more on how the climate change will have affected in the past and could affect in the future. And well, I'm glad to mention as well that he has been collaborating with several Mexican colleagues, mostly Dr. Adrian Perez Crespo and his students on these kind of studies, and that has helped to improve the work we are doing in Mexico. So without any further delay, I will introduce Florent. Before that, I just want to acknowledge the people from the National Coordination of Anthropology, and especially those focused on the information technology, Salvador Cristobal Jesus Portilla, Luis Olaf Ortega, and Juan Carlos Carrillo, who made possible both this transmission by Zoom, but also the YouTube at the channel of the coordination. And also we thank Eduardo and I to the head people at Centro, INAH, Morelos, and Subdirección de Laboratorios y Apoyo Académico for support this project. And well, without any further delay, I would like to give the voice to Florent. Thank you, Florent, to be here.
Thank you. Buenos dias, good morning, good afternoon, good evening, wherever you are located. I will present this talk about feeding ecology of fossil proboscideans, and basically it's a review of some work that have been published in the past years. Proboscideans have, as you know, a very rich fossil record all over the world that provides very useful insight into their evolutionary history and ecology from the late Paleogene to the Quaternary. And these species are distributed across the world and evolved in unique adaptations in response to changing environmental conditions. So understanding their diet is very relevant to reconstruct ancient ecosystems and also to assess the impact of climate change over time. Among the different techniques that we can use to reconstruct paleodiets, dental wear and stable isotopes are the ones that provide very relevant data. And they also offer complementary perspectives to reconstruct diet, habitats, and behavior. Dental wear, which includes two different techniques, microwear and mesowear, I will come back on that a bit later, will provide insight into diet and feeding behavior of proboscideans, and more generally can be applied also to all mammals and even to all animals with teeth, because we look at these patterns on dental materials. The microwear pattern are observed on the teeth at a high magnification between 30 to 1000, and microwear will reflect the last few meals of an animal. While mesowear, it's more a long-term proxy that study macroscopically the occlusal surface of the teeth and will reflect long-term dietary habits, maybe an average of diet over the last few months or few years. And these methods allow us to reveal if these species were grazing, browsing, or mixed feeding, so different categories of diets. And the integration of these different proxies allow us for a comprehensive reconstruction of proboscidean feeding ecology. As I told you before, microwear and mesowear record different timescale. Microwear, the diet at the time of death, mesowear, an average, a broader context. So combining the two, we can get access to different moments during the last years or days of life of an individual. And many studies have shown that how these methods used together can provide some very interesting results about ecological interactions in past ecosystems. Very often, these techniques are also combined with stable isotopes, which also provide very useful data, because they are also depending on the part that are studied, if it's bone from collagen from bones or hydroxyapatite from teeth, provide also insight at different timescales. So combining all these proxies provide a very nice reconstruction of proboscidean diets. In this presentation, I will focus on the chronological and geographical areas where most data were available up to now, which are the Pleistocene. It's where really the most studies have been done, and especially from Europe, the Levant, and also North and South America. These are the localities where I could collect data. So either my own research, or even, of course, very often with colleagues, and also data published in the literature by other scientists. So these are the localities from Europe. As you can see, they are mostly located in northern and central Europe. These are the localities from North America, so mostly in the US, but also there are, as Joaquin said before, many works done by Víctor Adrián Pérez Crespo, by Víctor Manuel Bravo Cuevas, by Alejandro Marín Leiva, on many localities from Mexico. So also in Mexico, the research in microwear is also very well developed. These are the localities from South America. As you can see, it's very low. So much more research must be done in this area. And these are the localities from the Levant. This is from a project that I started a few years ago with Rivka Rabinovich from all the localities from the Pliocene and Pleistocene from the Levant. Now I will introduce these two techniques that we use to reconstruct paleodiet. The first one is dental mesoware. As I told you, it reflects diet over a long period of time. In that case, we looked at the relief of the occlusal surface. So this can be applied to different species, bovids, cervids, camelids, and so on, equids also. But in the case of mammoth, it's a technique that was published by Juha Saarinen in 2015. And Adrian Lister also participated to this study, and they looked at the relief of the occlusal surface, and they measure the angle between different enamel bands. And depending on the enamel, if the enamel, depending on the angle, if the angle is more open or more closed, it reflects different types of diet. So more browsing or more grazing. It's also related to the proportion of C3 or C4 plants in the diet. This is an example from the work that was published 1 year later by Saarinen and Lister, that record different species of different samples of mammoth from the Pliopleistocene to the end of the Pleistocene, and that they shown that how mesoware can reflect diet. So you can see that some species were mostly browsing, like Mammut borsoni, or other ones more grazing, like Mammutus trogonteri, for example, or Paleoloxodont. You can also see that there is a very high variability in some samples, like in some species like Mammutus meridionalis or Mammutus trogonteri. There is a very wide variability in diet. So some, depending on the individuals or the localities, some are more or less grazing. This is another very interesting study that was published by the same authors, by Saarinen and Lister last year, that show how the evolution of diet of Proboscideans over the last 26 million years in East Africa. And so it's a huge amount of data. It's a huge work they've been doing, that also show how diet is changing through time and how this is correlated with climatic change and also with morphology. So in this case, you can also see this trend toward, so the scale is starts from 26 million years ago to the right, towards the left to the late to the Pleistocene. And you can see how diet is slowly increasing from more browsing to more grazing in the Pleistocene, but also you can see this very wide variability in diet. So also depending on the species, on localities, all that show this very wide flexibility in diet of Proboscideans. The second technique I will present is tooth microwear. So in that case, we look at the same wear, but at a microscopic scale. In this case, the microwear is due to all the abrasive particles that are consumed by animals. In the case of herbivores, it's the phytoliths that are present in all plants. Phytoliths are small siliceous microcrystals that are present in plants, but they are more abundant in grass than in other plants, than in ligneous plants, for example. And these phytoliths will create microwear patterns at the surface of the teeth, especially on the enamel, which is the part that we are studying. And other particles like exogenous dust or grit that can be present on the surface of the plants, or also if the animals are feeding close to the ground and can ingest some grit, some soil, will also produce microwear. But it's possible to differentiate microwear from diet than microwear from exogenous parts. Which is also interesting with microwear is that every time an animal is feeding, new marks will erase the past one and replace them. So there is a very fast turnover, which is about in a range from few days to few weeks. So what we can see with microwear is the diet of the last few days or few weeks before an individual death. So this will record really like a picture, an instantaneous of the environmental context of this population. And this is very interesting because we can also detect seasonal changes in diet. This is an example. Sorry, the text, I can now see the text in Spanish. Sorry about that. It's two pictures of an enamel surface of a monkey, a laboratory monkey, that was fed during few days. And these two pictures were taken using a scanning electron microscope from the same area, but 3 days apart. So on the left, you can see the initial picture, and on the right, 3 days later. And you can see that in only 3 days, how the diet is changing. Or the microwear is changing. You can see that new marks, those indicated by N, new marks appeared, some disappeared, those indicated by a O. On the left side, you can see that these huge scratches, these huge marks are gone 3 days later. So you can see that in only 3 days how the microwear is changing. To study microwear, we have to produce duplicates of the surface. So we create negative molds using dental silicone, and then a positive cast using epoxy resin. Then we analyze it. So there are different techniques. The one I use is using a stereo microscope at a magnification of 35x. And we analyze a very specific surface of the tooth and quantify different type of scratches. In the case of small animals like ungulates, in general, we sample all the surface of the tooth. In case of Proboscideans, due to the size, of course, we sample a small area on the occlusal surface of the tooth. So one cusp or few lines of enamels in case of mammoth, for example. And then we can quantify the numbers of features on the surface. So this, to make it simple, are two types: scratches. So these are these elongated lines these elongated marks on the surface, and pits. We can be small pits, large pits, depending on the size. So combining these two variables, we can differentiate between diets. On the right, you can see a bivariate graph. It's simply the number of scratches on the x-axis and number of pits on the y-axis. And these ellipses are a database that was published by Solunias and Semprobon in 2002 that show diet of modern ungulates, including, well, ungulates and Proboscideans. And it shows how we can differentiate between browsing diets, so feeding on leaves from trees, shrubs, and other plants, than diets from grazers, animals feeding on grass, principally. And then in between these two, the mixed feeders that can feed both on grass and browse. Either opportunistically, they eat both type of plant on the same day or at the same time, or seasonally, animals that can switch or shift from grass to browse depending on the season. And also fruit browsers that have a very specific microwear pattern, because they have puncture pits, which are very large pits and very deep. So this is characteristic of fruit browsing due to the consumption of seeds and other parts of the fruits. So now I will make a quick review of different species of mammoth, starting from the most primitive to the more derived, and we start with the Mammut americanum. So the American mastodon, it's a brachydont species, but it has very low crowned molars. And in this case, these are samples from mainly from the US, from different localities in the US. And this comes from two studies, one from Jeremy Green that was published in 2005, and one I did a bit later with some colleagues. And you can see that all populations fall mostly in the leaf browsing area. So all these mastodon were leaf browsers. There is some variability between sites, and also the intra-sample variability can change also, like those from Florida, indicated by the FL, have a low variability, and those, for example, from the Big Bone Lake can show a wider range of scratches, for example, so more variability between individuals in the same population, but all fall within the browsing area. Some of them also have puncture pits, which indicate some fruit browsings. So they were feeding on leaves from trees or shrubs, but also including fruits in their diet. Another mastodon, this one is from Europe. It's Anancus arvanensis, which also have very similar molars, low crown, so brachydont species. And in this case, three of the sites we could sample. So two from the two from the UK. So it's Norwich crag and red crag. One from France, Chiac, and one from the Netherlands. Show mostly browsing adaptation, except the Norwich crag, which falls in between browsing and grazing, so it could be a mixed feeder. In the case of the browsing ones, we also have specimens with puncture pits, which also indicate maybe some ingestion or the consumption of fruits together with leaves from plants. On this graph, I grouped together different species of gomphotheres. Now we move to South America. There are not so many data using this technique. In that case, I plotted samples from Brazil, Chile, Mexico. In this case, it's the data published by different researchers in Brazil, Chile, and Mexico. And you can see that there is quite some differences. If we look at the Notiomastodon platensis, that we heard about a few weeks ago with the talk of Dimila Mote, we see here that some samples were plot with the leaf browsers, other plot among the mixed feeders, like the one from Mexico. And also I plotted one data from Cuvieronus from Brazil that plots above the grazer. So there are also some variability also among the gomphotheres. In these studies, what is interesting is that they combined, or in two of these studies, they combined the dental wear with the study of plant microfossils from dental calculus. So extracting these plant remains from the dental calculus. And what is very interesting is that the plant microfossil support the dental wear data. So we have a good correlation between the two proxies. Now I want to present the changes in diet in the mammoth lineage. So from the Mammutus romanus to the Mammutus primigenius. So here we will now look at an area that includes Europe and the Levant. These are data from this mammoth. It includes data from red crag in the UK and also from Erkelamar in Israel. And all the samples, so in that case, each dot, it's one sample, one individual. So in that case, all these individuals plot within the leaf browsing, which makes sense because these species have a very low crown teeth adapted to feed on browse. So in that case, all these samples plot within this area. We could also look at mesoware in the case of the sample from Erkelamar that I studied with Rivka Rabinovich. And in this case, also the mesoware indicates with this quite low angle a most browsing diet. So the two proxies indicate the same tendency in diet. The next species that we have is Mammutus meridionalis. In this case, you can see that each one of these samples, most of them come from the British Isles. We have one from Netherlands, Oosterschelde, and then one from France, Chillac. And in this case, most of the samples fall either in the browsing area or in the mixed feeding, and then few of them at the edge of the grazing area. So already in Mammutus meridionalis, that have increased a bit in ipsodoncy. It has more higher crown teeth, but still not as much as, of course, the more recent species. In that case, you can see that we can see a quite large diversity in diet. So there is a quite high intraspecific variability that covers almost the full dietary spectrum, even if grass is still poorly represented in its diet. The next species is Mammut trogontheri. In this case, you can see that the diet, well, the samples are shifting slowly toward grazing. So we still have few browsing samples. We have more grazing and many samples that fall in between in the mixed feeding area. So in this case, we still have this large intraspecific diversity, but the diet is slightly shifting toward more grazing. And finally, in Mammutus primigenius, so the woolly mammoth, most of the samples fall within the grazing area. There is only few of them, like some localities from England, Crayford, Erith, and Sledgerin, that indicate more mixed feeding. But in the case of Mammutus primigenius, the diet is clearly dominated by grazing. The woolly mammoth has been widely studied also with using stable isotopes. So this is a compilation of different studies that was published by Hervé Bocherens a few years ago, and that show that in different ecosystems from Alaska, Yakutia, and Europe, the mammoth has always very high nitrogen values that also indicate this consumption of grass. So also in this case, microware and stable isotopes are very complementary because they allow us to get a very accurate idea of this diet. At the same time of Mammutus primigenius, we can find in some localities Paleoexodont antiquus. Here we have a very large amount of samples taken from different studies, and you can see that Paleoexodont covers the full range of diet, so the full spectrum from leaf browsing, like in some localities in Romania, Stenheim in Germany, Holon in the Levant, or Revadim, to purely grazing, like in Megalopolis in Greece, or Gesher Benodiakov in Israel. So, and also many samples that falls in between. So Paleoexodont was very flexible. So depending on the diet available or what he could find, adapt very easily to different habitats. What is interesting is that this is a study that Rivka Rabinovich and myself presented in the last Mammoth conference, but the data are still unpublished. You can see that in the same area, so in the Levant, we have some populations that are more browsing, like those from Holon or Revadim, and the other ones that are almost exclusively grazing, like those from Gesher Benodiakov on the right of the graph. This is just to show you that the difference compared to other samples. So samples from more Western Europe and samples from the Levant. So, but in the two areas, so either in Western Europe or in the Levant, we have the same range of variability for that species. So it's not only something that occurs in specific regions, but it seems it's a pattern that can be observed in different regions of where the straight-tusked elephant was present. In the case of these three localities from the Levant, we could also look at differences in mesoware. So we get the same result. The populations from Holon and Revadim more browsing, and the one from GBY here both browsing and grazing. Okay, so in the case mesoware indicates a broader range than microware. So there is a slight difference here, but the data are very similar. It must be in this case a difference due to the time frame of each of these proxy. Now, mesoware more long-term proxy, while microware is the most short-term proxy. For Paleoexodont, as well as the mammoth primigenius, was studied by stable isotopes. This is the same study by Hervé Bocherens that shows also the same pattern with Mammutus primigenius. I don't know if I can show. Well, Mammutus primigenius, which is located on top of this triangle, that indicates more grazing in open habitats, while Paleoexodont is present both in the upper part of the graph, but also in the lower right part of the triangle. So in that case, you can see that these two proxies are very well correlated, microware and stable isotopes. Finally, because next year the conference will be in Mexico, I could not avoid talking about Mammutus colombi, which is present in North America. So here I could found in the literature different various samples published from the US and from Mexico. In that case, the samples. What is interesting is that the samples from the US, so plot all with very high values in pits, while those from Mexico all plot with lower values and all in the grazing area. So Hidalgo, Oaxaca, Puebla, and other localities, all of them were all these populations were animals that were grazing. So also in Mammutus columbi, we have this with this very large intraspecific variability. So from populations that are mostly browsing to some that are exclusively grazing, and of course some in between showing some mixed feedings. Sorry. Mammutus, in some parts of the US, Mammutus exilis Mammutus columbi got stuck in some islands on the coast of California and evolved in a very small animal, so a dwarf mammoth that was present in three islands, Santa Rosa Islands, we call the Channel Islands in California. And in this case, the diet is clearly dominated by browsing. So there is a clear difference from Mammutus columbi that in this case indicates purely browsing adaptations in these isolated populations in these islands. So finally, this is the big picture combining all the samples we have described before. So what is interesting here is that the most brachydont proboscidean fall within the browsing area. So all these very low crown proboscideans so adapted to browsing, while the most ipsodont species show this very wide diversity of diet. We could expect that the most ipsodont were exclusively grazing, because ipsodoncy is supposed to be an adaptation to grazing, like what we can see in horses, through the evolution of equids from the Eocene to the Pleistocene. There is this increase in ipsodoncy and this shift toward more grazing. But it's not only a shift toward browsing, but more an expansion of the dietary niche and use of exploitation of browse and grass at the same time. This is also clear along the mammoth lineage. So from Mammutus romanus to Mammutus primigenius, we see this trend toward more grazings through time. So to sum up, the more brachydont taxa, so the gomphotheres and mastodont, were mostly limited to browsing, and this both in North America and Europe, with also very low dietary variability, while the most ipsodont taxa, so many species of mammoth, for example, or Paleoexodont, apparently consume the more diverse variety of plants. So there is more flexibility in diet for this species. So this high diversity of diet is very clear in Mammutus primigenius, and also in Trogontherae primigenius, and also in Paleoexodont. And as I said before, this increase in ipsodoncy through time is not only a way to shift the dietary niche toward more grazing, but it's this increase in ipsodoncy has the potential to expand the dietary niche, dietary breadth of a taxon, allowing this species to extend diet into the dietary range, but also still keeping the possibility of browsing as a potential diet. And there is still, even if you have seen that many studies have been done, there is still a lot to be done on paleo diet of proboscideans. So especially to integrate different proxies, so using dental microwear, stable isotopes, and other proxies like dental calculus to get a more comprehensive idea of proboscidean diets. Also, expand the geographic and temporal coverage of these studies. As you have seen, many areas are under studies. South America in particular, Asia and Africa. Also, more studies have been done in the recent past, but still many can be done in this area. And also going back in time, I focus on the Pleistocene, but microwear can be done on first. It's a non-destructive technique, and there is no limitation like collagen, for example, in stable isotopes. Microwear is preserved. If the preservation is good, it can be found in specimen in very old specimens. So I expand also the temporal range of this species. And also something that in my opinion, is very interesting is to explore interspecific interaction in these periods to show how this Proboscidean interacted with other contemporary fauna, and how these interactions have influenced or modified their dietary strategies and ecological niches. I would like to thank all these researchers that I've been collaborating in the past years. So thanks for their help in accessing these materials, and thank you for your attention, and I hope to see you next year in Mexico.
Thank you very much, Florent, and thank you for all the assistance to this talk. I will check if we have any question. Right away, I have a couple. One is how elephants behave in terms of the meso and microwear. What do they show?
Elephants, like modern elephants, or
Yes.
Okay. Yes, modern elephants are not. Well, there are some samples that were published by Surunias and Semprobon when they published the 2002 paper, and they are mostly in the browsing area, except the Loxodonta cyclotis. I think the forest elephant that showed also more mixed feeding, but they are mostly toward the browsing area.
Okay.
But it's a small sample, and I think also I think it would be a good idea also to sample more specimen of modern elephants, especially elephants that we know where they were living in the last days or months before they died. That would be very useful for us to get a more accurate modern sample for elephants and for Proboscideans in general.
I guess we could advise to Raman Sukumar to have those studies, because he seems to have a large sample of animals that they have collected within many years and dead on the field, so they should be able to carry on those. How about the yes, go ahead.
And these studies on modern mammals have been done on many taxa, but not on elephants. So yes, it's something that is really missing in this research. Sorry.
No. And how about the pygmy mammoth from the Wrangler Island? That has been controversy. These are different species of no, but how about the diet? Is there anything know about that?
From what islands? Sorry.
Wrangler, the one in north from Siberia.
To my knowledge, there is no studies on microwear or mesowear on these populations. As far as I'm aware, there is nothing published on those. But also, it will be interesting to see how their diet has been in comparison. For example, what happened in the Channel Islands in California. That we thought that there is a clear difference from the Mammutus columbi. So it will be interesting to see how they differ from the populations of Mammutus primigenius from the mainland. So from the mainland.
Okay. We have a few questions in the YouTube. First, you have greetings from Bea Menendez and Marta Benavente. Also, Oscar Sanchez from Tetenango, he asks, it seems clear that gnawing on abrasive grasses leaves occlusal scratches, but can occlusal pits somehow be linked to soft foliage or fruit consumption? Thanks.
Yes, pits can be related to mostly related to browse, and large and the very large pits to fruit browsing. So when we see very big and deep pits, so that they usually appear very dark, this indicates the consumption of fruits. And when the number of pits is very high, so above 40 in the area that we are quantifying, it mostly indicates browsing in dry habitats, so in arid or semi-arid conditions, because these are small pits, but they are very abundant on the surface. You can find about up to 100 or 120 pits, and these usually are related to the dust or grit present on the leaves of the plants in dry habitats. So due to the accumulation of dust in these dry areas. So if it does not rain, the dust accumulates over time and produce this high amount of pits. So we can see that, for example, in modern animals like the Mongolian gazelle, Tibetan antelope, many camels, dromedary camels, guanacos that lives in this type of habitats.
Okay. Victor Manuel Bravo congratulates you, and he asks, the mesoware angle method could be applied in mastodon teeth?
I would say yes. It would be interesting to look at that. With mesoware, one has to be very careful, especially in mastodon, to the wear stages, like to the ontogenic age of the individuals, because in mammoth, I think it's not a big deal, but in mastodons, the age could influence mesoware. So make sure that we take the same age class to record mesoware.
Okay, thanks. Fernando Tamariz Carrera says, the fact that microware of specimens of Mamurus Colombia in USA are much closer to browsing, could this be a factor for Mammut americanum to migrate further north in order to evade niche competition?
Yes, it could be. It could be something that we did not study, but it could be related to niche or to competition for the same resources. And that way, in my conclusion, I said that it's something that really needs to be done. In many cases, we focused on Proboscidean, but I think it's always interesting, like it is done with stable isotopes, to sample the full community to get the niche of each species and be able to formulate hypotheses about niche competition and changes in distribution of the species. Yes.
Okay. Oscar Sanchez, who asked the first question, he says, or is just the absence of scarcity of scratches that indicates more browsing than grazing?
Yes. Basically, the scratches indicate the abrasiveness of diet, so more or less grass in the diet. So yes, when there is very few scratches, it indicates browsing.
Okay. I have one more question, and this relates more to what currently is the vegetation in Mexico, that we have many areas with these spinous plants, cactaceae, agabaceae, and there has been a problem to figure, at least in terms of isotopes, what kind of signal will will identify that kind of plants. Is there anything related to meso or microware?
My,
Plants with big spines or
to my
so forth?
knowledge, no, there is nothing done on this type of plants. We would need teeth from specimen from animals that we know that in the last few weeks or months have been feeding or exclusively or almost exclusively on this type of plants to see what kind of micro pattern it could produce. But yes, it's something that have not been done. So if you can find animals that are that would accept to feed on these spinous plants for a few weeks or few days, a few weeks or few months, it could be done.
Thank you very much. I think we don't have any further question, so probably we can round up here. Thank you very much, Florent, for your participation. I think this is a quite interesting topic that certainly can give us a lot of information. And as you mentioned, just getting the overall picture with not just Proboscideans, but all the different taxa that we are able to study can provide better. We have a last one. We have a better chance to reconstruct, to make those paleoenvironmental reconstructions. And we have here one more. Can you illustrate? Let me see. Can you illustrate more about the possibilities to exploring interspecific interactions? For example, what could tell us about comparing or study together Proboscideans and other animals? From Yvette Espinosa.
We could have a good idea of the niche, the dietary niche occupied by each of these species in the same community, and especially not only from one community, which still is interesting, but if we could compare different communities through space or through time, we could see how these communities are occupied. Well, each species within the community can occupy different niche. Like if one species is able to displace the niche of other species or not. So I think it's not only the study of a single community, but comparing different communities on the geographical scale. So populations from at different latitudes or different altitudes, how this can change, and also with what the chance we have to be able to study the fossil record, take into account the temporal scale, to see how these niches are changing through time. So I think these are the two main aspects we could look at.
Well,
I hope I answered the question.
Well, thank you very much, Florent, again, and we thank to all the assistants to this talk. We remind you that they are kept on the web page of the seminar, and I think Eduardo could put the link for that in the YouTube page. And thanks again to all of you assistants, and thank you again to all the people from the National Coordination of Anthropology for your support in this session. So we look forward to meet you next week with our last talk on this symposium. And certainly we all are invited for next year, actually between September 7 and 12 to the 9th International Conference on Mammoths and their Relatives. Thanks for today and have a good day. Thanks, Florent.
Thank you. Bye.
Instituto Nacional de Antropología e Historia.
Secretaría de Cultura.
Gobierno de México.